you must be wondering that what Mila Jovovich is doing in this particular topic. Actually, if you go to all the zombie movies, and if they're good ones, usually they start from a bio-based laboratory where ultimately a vector, usually a virus, is released into the environment and infects the population at large, turning them into zombies. Now, we don't know much about the zombie part, but one thing is true, that there is a huge risk of some sort of virus getting out of a laboratory and ultimately infecting the population at large. Therefore, we try to develop strains and vectors which cannot affect human population. One such particular system is a baculovirus insect cell expression. Now, let's see that how does baculovirus work and why does it safe. So baculovirus infect invertebrates only. They can enter mammalian cells but cannot replicate. So when we are talking about invertebrates, what are the common hosts? Its common hosts are given in the images here, which is a lobster, a moth, a mosquito, and a fly. Its genome is circular, around 130 kilobase pairs, and there are two types: nucleopolyhedroviruses, which are most commonly used, and granuloviruses. Okay, and out of these, the most common is Autographa californica multiple nuclear polyhedron virus. And why it is important? Because from this army worm, from its ovaries comes a cell line, which is known as SF9, SF21, and one industrially developed Hi5, which is usually used. So basically, you are not rearing insects in the laboratory as such. We sometimes do, like Drosophila melanogaster, because it's studied as a disease model. But for this particular case, you have cell lines which you have developed from the ovaries, and ultimately SF9, SF21, and Hi5 are the most common ones. Its advantages are it gives you huge protein yield, very nice protein yields, 30 to 500 milligram per liter. Another reason for using insect cells, and sometimes they are even preferable over mammalian cell lines, because they do not require carbon dioxide, which are required by mammalian cells, and they can be easily adapted to high density suspension cultures for large scale expression. Usually when we are talking about other cells, you cannot grow them in liquid cultures at all. Viruses takes different forms. They can ultimately come out as a budded virus, but when they come out as a budded virus, what you can see here is that they are covered by an envelope. So if one particular virus, ultimately its nucleocapsid becomes enveloped by a membrane of an infected cell, but sometimes they are able to make this huge polyhedron, and that's what we are interested in when we are talking about biotechnology. Why we are interested in that? Because ultimately they make these sort of occlusions in which you have a lot of viruses, and they are secreting this. You see this gray area around? So and this whole gray area. So this gray area is basically a polyhedron mesh which is released by these viruses. Just look at the size difference. This is 50 nanometer and this is 5 microns. So now, for example, if you use polyhedron promoter and ultimately make them produce your protein, then you can see that how much protein you can produce, which would be packed in this 5 micrometer polyhedron structure. Also, you have to keep into account GP64, which is the protein basically which is involved in infection. Budded viral forms are less infectious and occluded forms are infectious. The promoter for the polyhedrine gene is exceptionally strong, which is good for the production. And transcription from this promoter can account for as much as 25% of the mRNA produced in the cells. Now, this is way higher from what we have seen in yeast, and and being a eukaryotic culture, this is very, very good. Moreover, the polyhedrin protein is not required for virus production. So it's a huge advantage that if polyhedrin protein is not even being produced, virus can still ultimately make a shell in which you can load your proteins. So this is very nice that it's not an essential gene. Replacement of the polyhedrin gene with a coding sequence for a heterologous protein. Heterologous protein in this case is the protein you want to produce, followed by infection of cultured insect cells, results in the production of large amounts of heterologous proteins. This is exactly what I have said, told you earlier, कि आप polyhedrin gene निकाल के उसकी जगह आप अपनी protein का gene डाल दें. It will produce copious amount of protein, and it's a very high volume expression system.